I have now. Hi, everyone. Welcome to the Research Innovation Spotlight Series, brought to you by the Alliance for SoCal Innovation and Bioscience LA, and generously sponsored by Alexandria Venture Investments. This is our spotlight on rare and orphan diseases. We have 4 distinguished speakers here to talk about their research, and we'll talk about them in just a sec. This really the point of this whole series is to connect all the great research going on in the labs and universities throughout Southern California, and connect them with the industry, so investors and corporate innovation folks, and just to make sure that we are pulling everyone together and understanding what's out there. So this is a really amazing ecosystem, and it's our job as the Alliance to pull everyone together and make sure that these connections are made. With that, I'm just going to do just a quick little bit of housekeeping and hand over to our esteemed moderator, Hallie Kuhn from Alexandria Venture Investments. What we're going to do today briefly is everyone's going to introduce themselves. Just please kind of keep it to your name, your title, and the organization you represent. We're going to skip the presenters for right now because they just get introduced by Hallie before they go and present. And the general format will then be each presenter will go for about 10 minutes of presentation time, 8 to 10 minutes, more or less, and then the rest of the remaining then 10 minutes or so of Q&A. Hallie will lead that Q&A, but so you folks, if you have questions, please do either raise your hand using the Zoom feature, or throw it into the chat, whichever works best, and Hallie can comment more on sort of what works in that regard, and we can keep the conversation going. But it should be a nice dialogue, and we hope everyone can jump in, and everyone should feel free to ask questions and get the learn more, investigate. So I will now introduce Hallie. Hallie Kuhn is the Vice President of Science and Technology at Alexandria Real Estate Equities, and also at Alexandria Venture Investments, which is the company's strategic venture capital platform. Actually, one of the most prolific biotech and investment biopharma corporate investors by new deal volume for the last 4 consecutive years, actually. Hallie has a PhD from Harvard University in systems biology. She was a Churchill Scholar at Cambridge University, recipient of a National Science Foundation Graduate Fellowship and National Defense Science and Engineering Fellowship, and a Harvard Ashford Fellowship, and has a BS from Harvey Mudd College. That's great, and has, of course, lots of actual also corporate experience and other things like that as well before joining Alexandria. So thank you for leading our conversation today, Hallie, and I will hand it over to you. Just to say hi, at least.
Yeah, my pleasure. Hi, everyone. And Steve, were you going to go through the rest of the room, or do you want me to jump right into it with
I will. I just wanted you to, yeah.
Okay.
It felt weird to introduce you, not actually introduce anybody else. So, okay. So now I'm going to call on each individual. Actually, first, before I do the individual attendees, I want to call on Dave Whalen, our partner in crime at Bioscience LA, who is our amazing collaborator who brings the life science expertise and knowledge to this series. The Alliance, we are sort of agnostic, but Dave really is the one who's deep in the trenches of understanding who's doing what and what's interesting and cool and should be known. So Dave Whalen, why don't you say hi?
Perfect. Thank you, Steve. Thank you, Hallie, and great to see everyone. Some familiar faces, some unfamiliar faces. Dave Whalen, the CEO of Bioscience LA. We are, as Steve kind of teed up, the independent kind of ecosystem catalyst, helping to grow life sciences, and in particular innovation around life sciences across the Southern California ecosystem, growing funding, growing space, growing talent, and really bringing it all together through messaging to grow awareness. Thrilled to be collaborating with the Alliance on this. Looking forward to hearing the talks. And I will just put in a quick plug, which I'll put in the chat, that next week is LA MedTech Week, and there's a whole variety of events going on. And if you go to lamedtechweek.com, you can take a look at that, and happy to share more in the chat or share more in email. So I'll put my email in the chat as well. Thanks, Steve.
Oh, great. Thanks, Dave. And yeah, in general, it'd be great to everyone connect. If you want people to connect with you, please put your email in the chat, and because otherwise we don't tend to share lists of attendees with everybody else, just because it tends to be problematic for some folks. So if you would like to get connected, please put your email in there, and we can then distribute that. That's like your sign to me that it's okay to share. I will be sharing all the presenters though. I think that sort of assumed you will all have their contact information. Unless of course you don't want to, let me know. Very good. And Andy Wilson, our executive director for the Alliance, why don't you say hi as well since you're here?
Yeah, I'll keep it short. Once again, Andy Wilson, I'm the executive director of the Alliance for Southern California Innovation, and we think Southern California is the center of the universe with respect to breakthrough technology, and certainly biotech and life sciences is an area that is certainly great legacy in San Diego, but also with Dave's help, really becoming an area of focus in greater Los Angeles. So I love sitting in these conversations because it blows my mind. And once again, thanks all for participating.
All right, thanks, Andy. So what I'm going to do now is basically go around the room. You know, it was Brady Bunch style. I know it's Hollywood Squares style. I will call on you, and then I will also tell you who's on deck next, so you can have a second to prepare. So in my magic window, actually, Jessica, you are up, and then on deck would be Michael Liebman. So.
Hi, everyone. Jessica Barnett with Bioscience LA. Excited to see you all here today. So thanks for attending, and I hope you enjoy the program. Thanks.
Thanks, Jessica. All right, Michael, you are up, and after that will be Pascal.
Michael Liebman, Managing Director of IPQ Analytics, actually on the East Coast in Philadelphia. We do disease modeling, and we're interested in pregnancy development and early disease, rare diseases.
Thank you. All right, Pascal, you are up, and next is Clace. Did I pronounce that correctly? Okay.
Hey, everybody. I'm Pascal Curti, and I'm a Senior Associate with Riverbest Venture Partners. We're headquartered in St. Louis, but I actually work out of our San Diego office. We're focused on biopharma and med device investing.
Fantastic. Thanks, Pascal. Clace, you are up, and then David Crean, you are next.
Hi, Klaus Hornstrand. Nice to meet you all. I'm a CEO of Wildwood Therapeutics. It's a seed-funded stage biotech startup funded by Westlake Village Biopartners here in lovely Thousand Oaks. This is my backyard behind me here.
Thank you, Klaus. I'm sorry for butchering your name. I got it now. Awesome. All right, David, you are up, and next is Ying Hung.
Good morning, everyone. David Crean, Managing General Partner for Coast Bioventures. Good to meet you. Good to see everybody.
Thanks, Dave. All right, Ying Hung, you are up, and next is Chris Jones after that.
Hi, my name is Ying Hong Gao, and I'm a venture partner of Viva Bio Innovator, part of Viva Biotech. We have about 80 portfolio companies in therapeutics, and many of them are in Southern California. Nice to meet everyone.
Thank you. Okay, Chris, you're up, and then after that, Lauren.
Hello, everyone. Good morning. I'm Chris Jones. I'm the Vice President of Business Operations at Wildwood.
Fantastic. All right, Lauren, you are up. Thank you. And then after that is Sonia.
Hi, everyone. I'm Lauren Bradley. I'm the Associate Director of Corporate Engagement at the Jackson Laboratory, which is actually based in Bar Harbor, Maine, but I am located in Los Angeles. Great to be here.
All right, thanks, Lauren. All right, Sonia, you are up, and after that, Martin.
Hi, everyone. I'm Sonia Satayesh, and I'm the Co-Director of Innovation and the Strategy Lead for Nucleate. We are a startup activator program where we focus on sourcing deals and working with the tech transfer offices across the three LA academic institutions of USC, UCLA, and Caltech. Glad to be here.
Thanks, Sonia. All right, Martin, you're up, and after that, Paul.
I'm Martin Broom, Corporate Collaboration Manager at the Office of Technology Commercialization at Children's Hospital Los Angeles. And I'm excited to hear the speakers today, and rare diseases are something we care a lot about at Children's Hospital. Thank you.
Thanks, Martin. Okay, Paul, you are up, and after that will be Kichiro.
Hey, good morning, everybody. I'm Paul Klikotka. I'm a Senior Medical Director here at Eli Lilly at our Biotechnology Center down in San Diego. And I'm really excited to hear the presentations today. So thanks for having me.
Great, thanks. Okay, thank you. And Kichiro.
Hi, can you see me?
Yes.
Okay. Hi, my name is Kichiriano. I'm from the Daiichi Sankyo Inc. I'm the director of the global business development. I'm very much looking forward to our presentations on rare disease today. We're focused on oncology, autoimmune diseases, any other focus. So I'm fine with open to any kind of discussion. Thank you.
Awesome. Thank you. And we'll probably get a few more people dribbling in throughout the presentation, but they don't get to introduce themselves. And Gigi, our newest member of the Alliance. You want to say hi, Gigi?
Hi, yes, I'm the events and marketing manager for the Alliance, just on recently. Thanks.
Thanks, Gigi. Great. All right. So I was premature before. Now I can officially really now hand it to Hallie, and who's been fully introduced. But Hallie, if you want to say anything more that I may have missed, please let me know. But great. Thank you all for being here. And Gigi, and Hallie, it's all for you. So, thanks.
Great. Thanks, Steve. And really nice to see everyone. We decided to sponsor this event after I took part in the targeted molecular oncology event that we had last year. And I really loved that it was a more intimate forum. So I just, as people have questions, really feel free to jump in. We'll leave time at the end, but I really want to make this as, kind of dynamic and engaging as possible. So, whether that's in the chat function or just, going off mute and jumping in with a question, please feel free to do so. So we're going to kick it off today with Dr. Alysson Muotri. He's the professor of pediatrics and cellular and molecular medicine at UCSD. His lab utilizes novel models, including brain organoids, to explore the complexities of the human brain, and in particular, some of the mobile elements that drive diversity during neuronal differentiation. And his work investigates, everything from the fundamental mechanisms of brain development, evolution, neural disorders, but also really has provided a unique lens into understanding, the etiology, but also potential treatments of rare CNS diseases, which he'll be discussing today. So I will pass it over to you, Alysson.
All right, thank you. Thank you for the invitation, and thanks everyone for attending. I'll go ahead and share my slides here. It should be seen like full screen. Real pleasure to be here on a Rare Disorder Day. I'm a professor of faculty here at UCSD. I'm also a father of someone with a rare disorder. This is my son, Ivan. And like Ivan, there are so many people who have neurological disorders. And one of the things that we see that is problematic is that some of those, especially genetic disorders, really starts in uterus. And that has been like a real challenge for neuroscientists to fully understand how the brain is formed and what can go wrong when you have, for example, a genetic mutation. So the inaccessibility of the in utero environment is really blocking how we can understand disorders and actually provide mechanisms of action. So the idea to use stem cells and create brain organoids or cellular models in the lab allow us to take that brain from the uterus and in our control condition of the lab really create these three-dimensional structures that self-organize. And what you are seeing here is just a dish full of these tiny brain organoids. Then they have like 2.5 million neurons in there, size of a bee brain, 0.5 centimeters in diameter. And they really self-organize on something that resemble the human fetal brain. And you can actually keep them for several years in the lab and studying how the different cell types emerge and what are the different mechanisms implicated in diseases. And I'm pointing here to the Zika virus because I'm originally from Brazil, so I got involved in this research really early on. And during the outbreak over there, there was really no evidence that the virus would be causing these microcephalic phenotypes that you see here in the screen. So it was only with the brain organoid technology, when we exposed the Zika virus to the organoid, that we saw that some of the intermediated neuroprogenitor cell was being killed, reducing the cortical plate, creating the macrocephalic brain. That could not be modeled in mouse. We know now why, because the mouse neurodevelopment is really fast. In 20 days, you have a full form mature brain, whereas in humans, it takes 9 months to actually get there. So that was a slow progression of human neurodevelopment that allow us to show causation. But most importantly, just 2 years after the outbreak, and as far as I know, this is a record time from causation to treatment, we were able to repurpose So fosbuvir, which is a FDA-approved drug, to block the vertical transmission from moms that are infected to the fetus. So now we know what to do, and we're all obviously screening for other drugs that could be even more potent or more specific to the Zika virus. So this is something that is the environment creating something to the organoids. You can visually see it, but the diseases that mostly I'm interested in the lab are diseases of network connection, autism, schizophrenia, or some of the rare disorders that actually affect how the neurons communicate. To do that, we really need a functional assay. And we've been optimizing how to make these organoids become functional by changing the media here and there. And we finally create something that seems very useful. And we analyze that by plating the organoid on top of a multi-electrode array dish, so we can follow the network activity through time or through years and see exactly how these networks evolve. And one thing that was a big surprise to us is that not only they seem to follow the human trajectory, but they become highly complex over time to the point that they release or they show some of these neural oscillations. These are highly complex network formation never seen before in an in vitro system. So they start at 4 months of age. 6 months, you have highly stereotyped networks. By 8 months, these neural oscillations become highly complex. Different frequencies emerge, and that's why we see like they start to overlap. So why this is important? Well, I mean, this is the first time that you can bridge the basic biology, because that's what we do in the lab. We look at synapses, gene expression, cellular interaction, but we are far from human cognition. By having a system that allows us to detect these neural oscillations, it really bridges what cognitive neuroscientists have been doing for several years, which is to correlate the different frequencies of the brain with human status or humans' behavior or diseases. So we are one step towards that goal, and that allows us really to have a better disease model in the lab. I highly recommend you guys to read this article. Our research was highlighted in the New York Times. I love the title, Organoids Are Not Brains, How Are They Making Brain Waves? And that touches about this next step in disease modeling. Also, you see that it raises some of the ethical and moral questions about the use of this technology. But that's another discussion. So now we don't have a cure or specific treatments for most of these neurological conditions. How can we take advantage of this model and really push forward to do something like that? And that's what my lab has been doing. This is the bread and butter of the lab. This is just a list of some of the diseases that we have. You probably realize that we tend to be under the spectrum or the umbrella of autism. So we're looking for social disorders and rare syndromes with a clear genetic mutation. And we create organoids from them. We also use isogenic cell lines when we can't find enough patients to test. And we use that to test drugs. If it's polygenic, sometimes we look for a compound that can rescue a specific phenotype. If it's a monogenetic disorder, sometimes we try to regulate the gene using ASOs or gene therapy. We are also using the technology to optimize gene editing, for example. Another thing that we are doing is really testing different delivery systems. We are really tight on the AAV9 serotype for delivery. So we are broadening up the different serotypes and testing them on the organoids, which has the relevant cell types for human nervous system, which seems to be like a bottleneck to move into clinical trials. For some of these conditions, we already entered clinical trials. Others, we are just starting or in the middle of the process of finding mechanism. I'll highlight some of the successful cases that we have here. This is a mutation in CDKL5. It's called CDKL5 deficient syndrome. So we create brain organoids from them. They mimic one of the most hallmarks, most severe hallmarks of the condition, which is the early onset of seizures. So these kids are born seizing after like a month or so. So organoids with the mutation, they have this hyperexcitability. So these neurons are firing way more compared to controls. And a single injection of AAV inside the organoid, we actually don't inject, we just add the virus to the media, and we are able to transduce about 10% of the neurons with the correct version of the gene. That was enough to push back this hyperexcitability to control levels. And you can keep following the behavior of these networks over time, and you see that that single injection actually was enough to inhibit the excitation to become some seizure-like signature. So this is very nice. Technology has been licensed by a company that's really moving forward with a potential clinical trial with this disease. The other thing that the organoids are really good is that because it's an in vitro system, I can play with the different cell types and I can mix and match different genotypes. And I'll give you like one example here that I think is really cool. This is Rett syndrome. Another rare syndrome caused by mutation in the MECP2 gene. What's important in Rett syndrome, or intriguing, it is that although the mutation is in all cell types in the brain, if you just restore the expression in astrocytes, you rescue the disease, at least in mice. So that opens the possibility that, well, I mean, maybe a gene therapy in astrocytes might be like something really cool. We're definitely exploring that, but it's very difficult. Another possibility would be, what if we just transplant normal astrocytes in the brains of these people? So we created that, or recreated that experiment using the brain organoids. In one side, we created the glial progenitor cells. On the other side, we have the organoids, and then we infuse the glial progenitor cells inside the organoids. They really spread out and take over, and they differentiate into healthy astrocytes. So then we ask what happens, and this is one of the really clear results that we have. If you look into normal neurons on with normal astrocytes, the astrocytes are in the background. I'm just highlighting the neurons so you can visualize the morphology here. They look really well, highly arborized. If you now work normal neurons with Rett astrocytes, as expected, the Rett astrocytes are bad and makes the neurons to become immature or not to develop. The same is true in the disease context. When you have both mutation in neurons and astrocytes, the neurons just don't thrive. But look what happens when you now move the mutant neurons and add them together in a health astrocyte context. You basically rescue everything. I'm showing morphology, but at the functional level, this is also rescue. Most likely, these health astrocytes are contributing to the synaptogenesis in helping the neurons really to form new synapses. So I'm a co-founder of this startup, Lyser Bio, who is really one of the few companies that can manufacture large quantities of iPS-derived cells. And we have this goal to really push that into IND by the end of next year. So just finishing here by saying that one of the problems that we have, most likely others have the same problems with rare disorders. Where are the patients? I mean, we're sometimes studying a gene and we have a drug or a disease that we need patients to study them. And this is true for physicians, companies, startups. So we thought about how can we help to really integrate everybody, and we decided to create an app. So this is called Tismo.me. It's an app that uses like machine learning and deep learning to really connect people with rare diseases. We initially focus is in a first phase 1 now for autism, but we can already search people with different characteristics, a gene mutation, or by profile. And the app is really taking over in the community for autism, and people are using that to stream, to advertise, to create courses, things like that. So we're really helping to create a new social network that is helping to connect those people. And for me as a scientist, with a click of an app, I can really find where are the patients with the mutations that I want. So it's been very helpful, and we have several projects already going on. So I stop here, but thanking like my funding agencies and of course my lab. I briefly described lots of research with lots of people, couldn't really give the credit to each one of them. Thank you.
Great, thank you so much. I'll open it up for questions if anyone wants to jump in. Otherwise, I can start with my own. Maybe I'll kick it off. You know, I was wondering, how do you think about, so you have these organoid systems from kind of early developmental cells, right? And how do you think about kind of the window of intervention for various diseases, right? Especially for those that are, you know, even in utero, right, kind of potentially have neuronal impacts. And I know it's different per disease, but just would love to hear kind of your thoughts on that.
Yeah, that's a great question. So the organoid really mimics these embryo-fetal development, right? We're really capturing that stage. So that's great for proof of concept or to find mechanisms. But really what we are doing is pushing this technology to mimic more mature brains. So we could get like early postnatal, and I believe that soon we will be able to get and model something that will resemble a little bit later in development, like 2 or 3 years old. And we're doing that by systematically improving the technology, for example, add input and output to these organoids. We are also using different ways to really senesce the cells inside the organoids, so the gene expression profiling and the electrophysiological profile are more close to a more mature brain. So that would allow us to perform these experiments in a more therapeutic way. Yeah.
Yep. Okay.
I have a quick question. Can I ask a quick question? So
yeah,
I noticed that, and that's true for autism. Actually, it's probably true that for most autism we don't have an explanation, but most of your disorders were deletion, duplications. Are there specific challenges, you know, dealing with those types of disorders versus single point mutations? I know you gave us a couple of examples like CDKL, but I'd love your thoughts about that. I would have thought working with big deletions, duplications would be even more challenging, but maybe you can educate me.
That's another great question. And it is more challenged because now we are dealing with a polygenic condition. And what we are finding is that depending on the condition, there are driver genes, maybe 1 or 2 genes that really push the phenotypes or the most severe phenotypes. So that becomes like a target right away, especially on the duplication syndromes. I mean, if you find a gene that is really pushing the phenotype, you can target that gene with an ASO, for example. So these are the easy cases, but the most difficult cases are the ones where it's probably an epistatic interaction among all the genes that are involved on the deletion and surrounding regions of the genome. So these are harder to find single point therapies. And most likely we end up looking for molecular pathways that are affected and looking for pharmacological interventions. Yeah.
I guess maybe then another question on, as you start to kind of identify more mutations, right, for what were kind of originally kind of thought of as these polygenic diseases, you kind of get to this kind of N of 1 and ultra rare type diseases. I guess just, you know, how do we think about that, especially with kind of like the industry hat on, right, and translating some of these therapies? I know there was a great case study, it was out of Boston, you know, Mila's kind of therapy where they, you know, created an ASO for one individual. It seems challenging to translate, but would love just to hear how you think about kind of moving some of these potential treatments to groups of individuals where it may be, you know, a very, very select few patients.
Yeah, no, that's great. And I think, I mean, companies are interested on making some profit as well. So, I mean, the N equals 1 is really a challenge. And that's why we create the app. Even mutations in the same gene, I mean, it's hard to find other people. So the app is really helping us to connect with lots of people. I think one thing that is missing is really an effort to have people sequenced. So I interact a lot with families on the autism spectrum, and I can tell, I mean, phenotypes from them are different. So most likely they all have different mutations. But I would say that 90% of the autistic community has not done a whole genome sequence or an exome sequencing to try to find the causation of the condition. And I blame a little bit on the doctors that do not stimulate the sequencing of those people. But as we have more in the data set, I think we're going to solve those. But there's always going to be private mutations, and there's always going to be solutions for private mutations as well. And you mentioned Mila's case. It's probably one in the world, but at least, I mean, as a proof of principle, we know that that can be done. So the idea is really to use the technology and apply to different mutations. Sometimes it works, sometimes it doesn't.
Any other questions?
I would like to defend the doctors. I would like to defend the doctors who don't bother doing the exome. Have you actually tried to get an exome on a patient who has no insurance or poor insurance? We spend more time, with companies trying to get authorizations. The good news is that now Medi-Cal patients are getting covered, but just very recently, maybe in the last 2 weeks or so. I have, a new company, I will get free exomes or try to be creative. They have these companies that do free panels. And if you do enough free panels, you'll get, maybe 1/10 of an exome, but it's very challenging. Unless exomes get cheaper, it is always going to be a challenge. I personally would like every patient I see, I'm a clinical geneticist, every patient I see have an exome even before they come to see me. I mean, that would be ideal, but I think we're a little bit way off for that.
Yeah, absolutely. Yeah, April, I see your hand up.
Oh, great talk, Alysson. Beautiful work. I was just curious how you mentioned you're doing AAV-based screens, is that right, in the organoids? Yeah. I was just curious how that is going in terms of, do you see that if you screen sort of standard AAVs, that it represents what we see in the adult cells, or do you see a developing AAV signature, or I'm just curious how that's going.
Yeah, so right now we can mature these neurons in the organoids. At least they behave like a little bit more mature neurons, and they already express all the receptors that they should have. So we believe that it matches more or less a more developed brain. And so that's why we decided, let's test the AAVs. And the AAV9, as I pointed out, we get about 10% infection just by adding the virus in the media, leaving there for 12 hours. But we already found like other serotypes where we can go to 9%, which is very encouraging. I mean, I was never expecting to find a serotype that would be that efficient, but they do exist. The question now is to see if they will have the same efficiency in a more adult brain. And I don't think it helps to test in large non-human primates, because the receptors and everything else is very different. So I think it's only when we move into a clinical trial that we're going to learn how they behave.
Yeah. Great. Well, thank you so much. And I think that's a great transition to Dr. April Pyle. So I'll go ahead and give a brief intro here and then kick it off to you. So Dr. Pyle is an associate professor in the Department of Microbiology, Immunology, and Molecular Genetics at UCLA, and is also a member of the Eli and Edythe Broad Stem Cell Center, the Center for Duchenne Muscular Dystrophy, as well as the Johnson Comprehensive Cancer Center at UCLA. So her lab uses multidisciplinary approaches to study human pluripotent stem cell biology, differentiation of these cells, and how that relates to both, basic aspects of stem cell biology, as well as how those translate to specific kind of disease types, particularly related to skeletal muscular diseases. So I will pass it off to you, April, to kick it off.
Great. Can you see my full screen?
So it looks like it's in presenter mode.
Okay. Double screens always get me.
Perfect.
Okay,
it looks good. Yeah.
Great. Okay. Thank you so much for giving me the opportunity to tell you about our work. I'm at UCLA in the Broad Stem Cell Center there, and my lab is really focused on using human pluripotent stem cells and more recently organoid models for looking at neuromuscular diseases. So thank you, Alysson, for that introduction and the highlight of the importance of pluripotent stem cells. So one of the things that we've really been focusing on is developing disease models in a dish for one of the most devastating neuromuscular diseases, Duchenne muscular dystrophy, which I think is really one of the kind of leading the way in a lot of preclinical and clinical based strategies. So it's really an exciting time in Duchenne. So Duchenne is a very devastating skeletal muscle wasting disease. Patients die typically in their teens or their 20s of cardiac and respiratory failure. And so we wanted to try and develop really a strategy to do better than the current standard of care, which is currently steroids. When we embarked on this project, although there's some exciting, therapeutic avenues ongoing in the field, which is really exciting. So in my lab, we, work with pluripotent stem cells. So we were able to develop pluripotent stem cell lines in the lab. And so our interest really has been to try and develop a strategy to actually restore the missing protein in this case, which is dystrophin. And so I'll tell you about some of our efforts in looking at this using direct in vivo delivery of a CRISPR-Cas gene editing strategy, as well as our approach in the lab has been to really try and develop cell-based strategies for neuromuscular diseases as well. So we've been making progress in developing skeletal muscle in a dish, and we're currently looking at the functional, potential of these cells in vivo in mouse models of disease. So some of you may know that Duchenne is due to mutations in the largest gene in the genome, dystrophin, which leads to out of frame mutations, which lead to no dystrophin protein being produced. And this is devastating for the skeletal muscle cell because this essentially disrupts the contacts with the muscle membrane and the extracellular matrix, which is key to stabilizing the muscle cell, as well as saving that dystrophin glycoprotein complex of proteins that reside on the muscle membrane. And so you can see that without this protein, that there's no stabilization of the muscle membrane. However, there's an allelic disease, and these patients come into the clinic perhaps in their 60s, don't even know that they have a muscular dystrophy. And this is called a Becker muscular dystrophy, where there's essentially an in-frame deletion in the dystrophin protein. And you can see below in cross sections of muscle that in DMD, there is no green. And so there's no dystrophin protein being made. But in Becker, again, there is some dystrophin being made. It's not identical to the wild type, but there is a lot of interest in developing therapeutic strategies for restoring the reading frame from a Duchenne to a Becker, which is one of the things that we were interested in thinking about. And so one of the interesting approaches that we embarked on was thinking about a hotspot mutation of region of mutations in patients with Duchenne, which is between exons 45 to 55. And so it turns out that about 50% of Duchenne patients have a mutation within this region. And so when we embarked on this project, we were concerned about developing a CRISPR gene editing strategy that will require developing guides for each patient mutation. And so we wanted to see if we could actually design a system that perhaps seems counterintuitive, but to actually delete the entire hotspot region in Duchenne muscular dystrophy, and to see if this would actually enable us to generate from a Duchenne to a Becker-like protein. And so long story short, this actually works really efficiently. So we developed a CRISPR gene editing strategy with guide RNAs actually flanking this hotspot region in the introns. And then the Cas9 protein will come and cut these intervening regions in the introns and actually piece back together the gene. And you can see here a reframing event rejoining exon 40 between 44 and 56. And so then when we differentiate our pluripotent stem cells into affected cell types, cardiomyocytes and skeletal muscle, you can see we have the ability to restore the missing dystrophin protein. You can see to the right on the Western blot that we've been able to restore dystrophin, and it's a truncated band, again, demonstrating that we've made this in-frame deletion. So pluripotent stem cells have been a really powerful model to be able to develop strategies such as this gene editing strategy. But the real challenge is, how do we translate this platform in vivo? And I think this is kind of a challenge for many CRISPR gene editing based strategies at the moment. And so we've been looking at many different ways to deliver the CRISPR machinery. We've been using AAVs. We've spent a lot of time developing and testing non-viral based strategies, as well as using the pluripotent stem cell derived cells differentiated into skeletal muscle and cardiomyocytes in a dish, and then using these as a tool to restore dystrophin by a cell-based delivery strategy. And so this is just giving you a snapshot of the efficiency of what we're getting by delivering the cell-based strategy. So here we developed a directed differentiation strategy to make skeletal muscle in a dish from pluripotent stem cells. And we use the corrected gene edited lines, and then we can actually engraft these directly into mouse models, in this case, an MDX mouse model in vivo. And you can see that we're able to restore dystrophin. So this is again shown in green after the CRISPR-Cas9 gene editing strategy, and also shown in red with the human cells. So we're able to efficiently generate skeletal muscle in a dish, which has been a challenge in the field up until recent years. And now we're able to show that we can use these cells to restore dystrophin in vivo. However, the challenge with this strategy is that direct cell-based deliveries in muscle, because muscle is 40% of your body, would require direct IM injections. And so for something like Duchenne, this currently is inefficient because multiple muscles are affected. So we wanted to think about other delivery-based strategies. And so to do this, Courtney Young, who is in the lab, developed a humanized model of Duchenne. So we actually took an existing DMD, humanized DMD model that was available at JAX, and we actually mutated exon 45. So we deleted 45. So now we have a human dystrophin gene in a mouse model that's mutated. We crossed this to the dystrophic model to be able to test therapeutics in a humanized dystrophic mouse model. And so when we do this then, and we try and deliver this both IM and systemically with an AAV-based strategy, we've tested AAV6 and AAV9. Currently, they work really well. And so you can see that after IM injection on the left, you can see dystrophin restored in a cross-section of muscle, as well as in systemic-based strategies. You can look at the heart and multiple muscles. You can see restoration of dystrophin, again, using our AAV-based strategy to deliver the gene editing platform in vivo. And below, you can see Western blots showing restoration of dystrophin protein in this context. So we're really excited about this strategy to, develop our gene editing strategy forward. And so we developed a company, Courtney, who I keep mentioning, has now spun out this company, Myogene Bio. We're currently raising a Series A, and this is to really move this forward for patients with Duchenne muscular dystrophy. And so we have a patent and exclusive license negotiated from UCLA. We're excited. We recently received a CERM-TRAN grant to move this forward, looking in dog models. So we're stepping this forward, and really excited to continue on this journey with my co-founders. And then I'll spend the last few minutes telling you about a new model that we've been developing in the lab, which is to try and use human pluripotent stem cells to look not only at the muscle side, but also the innervation of the muscle at the neuromuscular junction. And we're doing this for multiple reasons. One, because we think innervation is important for regulating the muscle. And so without this in our model of skeletal muscle, we don't have the full model. And so we're trying to build a better model, but we're also trying to develop this strategy to look at neuromuscular based diseases with a focus on the neuromuscular junction. So we've been adapting our 2D protocol to a 3D skeletal muscle organoid, and that's shown here. And so just at the top panel, you can see the general cartoon scheme of the protocol. And then below, you can see our ability to make a 2D based skeletal muscle system and translate it into a 3D based system. And so it looks like this skeletal muscle organoid system is working. In fact, we think it's pushing the skeletal muscle a little bit earlier. And also, if we look in the context of neuromuscular junction formation, we can start to see some of the signs of key neuromuscular markers, such as alpha bungarotoxin and and it's difficult to see, but zooming in there, looking at the white, you can see regions of neuromuscular junction formation. And so what we're really doing is trying to optimize this protocol to be able to develop disease models in a dish for ALS, in particular, because there are a number of both genetic and non-genetic ALS patients, and we want to be able to really screen through as many different ALS iPSC lines as possible with a focus on the neuromuscular junction. So we've been developing a high content screen with Robert Damaso in the UCLA Screening Center to look at neuromuscular junctions and to try and screen for drugs that save the neuromuscular junction and prevent neuromuscular junction loss, because any stabilization of the neuromuscular junction would really extend the lifespan of these patients. And so that's kind of where we're going with our ALS-based neuromuscular junction work. And so with that, I will just tell the thanks to my lab and our funding sources. So as I mentioned, this work has been funded, well, moving forward, work is funded as well from CIRM and NIAMS, and then many people have contributed to different aspects of the work, specifically Melissa Spencer and Courtney Young, who are my partners in crime in developing the CRISPR gene editing strategy forward, and Michael Hicks, who I showed some of the engraftment data, who now has his own lab at UCI. So with that, I thanks for your attention. I'd be happy to take any questions.
Great, thank you so much. I'll maybe kick it off with just kind of a mechanistic question on the CRISPR side. But what have you guys seen in terms of kind of, rate of indels and, in terms of kind of efficiency with the current CRISPR systems, and how much optimization was needed there to knock out kind of such a large piece of the DNA?
Right. That's a great question. I showed one slide of here it works, but in reality, we spent many years, identifying the best guides, the most specific guides, the ones that, where to put the guides. We have a strategy that we think is important in the guide positioning, and then testing off targets in the context of those guides. So we spent many years identifying the right guide pair. I would say that most efficiently generates this in-frame deletion. And then we've looked at candidate gene off targets, and we don't see any with the optimal guide system, but this is really important. And this is something that we're looking to move forward in the next phase to do whole genome analysis. But in our preliminary candidate studies, we don't see any off targets, and we don't see any gross rearrangements. So with the current pair of guides, it looks pretty efficient, but yeah, that's something that we're embarking on looking at in more detail.
April, it's wonderful that you and your group set up this company. Could you share with us some insights, what the process was, how difficult was it, and was it worthwhile doing it yourselves rather than engaging an outside company?
Yeah, so that's a great question. So I kept mentioning Courtney because she's a superstar. And if you don't know her, you should know her in the region. But she is the real reason why we moved this to a company, because she was a graduate student in the lab and this was her project. She has a cousin who has Duchenne, and she is on track and has been in the DMD research world for more than 10 years, even before kind of coming to us. And so she's a real powerhouse that wanted to move this forward translationally. So I think the timing has to be right. The project has to be right. The person has to be right. The team has to be right. All those things need to be in place. I wouldn't have done it had that team not been in place. Having Melissa Spencer on board, who has done clinical trials in Duchenne at UCLA, and then having Don Kuhn at UCLA, who is, one of our advisors, has really helped us kind of push this forward. And so UCLA has been very supportive. We were able to negotiate the IP on the platform. And so, now we're just doing the safety studies and the efficacy studies. And as I mentioned, we're looking to move this forward to large animal models. We are moving this forward with CIRM funding in large animal models currently in London.
Is there a reason why it can't go straight to humans? Do we need that large animal work?
Yeah, so we had a pre-IND meeting with the FDA, and these were kind of discussions that we had in terms of looking at safety and efficacy. And we ourselves wanted to look at this in 2 models, and in particular because the mouse models of Duchenne are not always the best models. And so because we're doing a systemic based delivery strategy with AAV in vivo, we wanted to look at this in another model to make sure to look at efficacy, safety. And we're also, I didn't talk about doing a strategy where we want to potentially be able to redose this platform if needed. And so we want to do a lower dose to be able to get a higher efficiency of dystrophin, and if need be, develop a redosing strategy. So that's something that we also wanted to do and look at in the dog model in more detail.
Maybe I will jump in.
Oh, go ahead, Ying-Hong.
Hi. I will jump in to ask your comment on this approach comparing with, for example, RNA-based therapeutics.
Mm-hmm. Yeah, so here we're causing a permanent deletion, right? And so in theory, we should be able to restore dystrophin permanently with 1 shot. And so then the strategy thus far has been with antisense oligonucleotides. These patients have to come in for weekly infusions. And then it's super transient and I would say very low effectiveness, although it did kind of pave the way for, beginning therapeutics in Duchenne. So it's very ineffective. And so a permanent in-frame deletion would then restore the reading frame in skeletal muscle. And the important thing about skeletal muscle is that it's multinucleated. And so in fact, in Duchenne patients, it's known that you don't need very high levels of dystrophin. And so even, the ASOs were approved at less than 1%. And so we're getting closer to, in the heart, upwards of 50%, in skeletal muscle, upwards of 20%. And so this strategy really is something that would be a permanent based strategy we expect, at least for the lifetime of those muscle fibers. And so I think this is an advantage over a transient based strategy, in my opinion.
So is it like once for lifetime cure?
Well, so currently, I think the most efficient way for a lifetime cure would be to target the stem cells, which we don't see targeting effectively using the current AAV-based platforms out there. And so I think in the end, we will need either a stem cell-based strategy in combination or a redosing strategy to be able to redeliver as the patients grow, their muscles grow. And so the question is, how long is that? So we expect a window of 5 to 10 years, but being able to redose them, we hope would be able to extend longer than 10 years.
April, you mentioned AAV 6 and 9. Are you using the new AAVs that are more muscle trophic, like AAV Myo AAV that have recently been published?
So, as you know, when you go down a preclinical clinical train, when you set that in motion, which ours has been mainly AAV9 because of the micro and mini dystrophins, we're doing trials in those. We have really been focusing on looking at this in AAV9, although we are testing other AAVs, and Melissa and I are actually looking for satellite cell specific AAVs and have a handful that we're screening through now. So we will probably second generation look at some of those AAVs, but right now AAV9 is what we're moving forward because of the current trials that are using that and have already been effective in Duchenne patients. And they target skeletal muscle and the heart really nicely. So that's what we're doing.
I had a question about the AAVs. So how are you, if you do need to redose, how are you examining immune inactivation and whether that's even possible?
Yeah, so we actually have a platform I'm happy to talk about in more detail, with the whole Myogene team, if anyone's interested. But we're actually using a cocktail of immune suppressants that actually was first developed in collaboration with Barry Byrne, who's actually doing this in some other rare diseases currently, although just direct IM, and we're looking at the potential of redosing in a systemic-based strategy. And so it's based on FDA-approved drug cocktails that enable us to redose, which is really powerful because this enables us to potentially lower the titer, which enables us to have a safer profile. And so, yeah, that's kind of the thinking at this point. And at this point, we're thinking about redosing. We're thinking about doing multiple injections up front just to get that level needed of dystrophin. And then hopefully we don't need to go back and redose later in life. But if we've used this strategy up front, then we have that opportunity later, and we think it's more ethical to try that. So that's the strategy we're using, and it will apply to other rare diseases in muscle. So I think that'll be powerful in not just Duchenne, but in other rare muscle diseases.
Are you looking at biomarkers or pharmacodynamic type responses? How will you measure that in terms of what's enough, right? How do you know that you've gotten enough into the target tissue?
Yes, absolutely. So we have been doing all of those. We've been looking at immune profiles. So Melissa Spencer is an immunologist who has been working with several folks looking at what is the profile, how do we know that, and we've developed kind of a screening system that allows us to see the efficiency of redosing and the immune response. And we've been doing single cell and other kind of analysis to look at which populations are shifting in response to these drugs. So we think we have a good cocktail that's working really effectively. But yeah, we're also testing the redosing strategy in dogs in the CERM grant.
Yeah, that's what the FDA wants to hear, I'm sure. So yeah,
that's exactly why we're doing it.
Yep. Thank you.
Yeah.
Thank you so much, Dr. Pyle, and it's a great transition to our next speaker, Dr. David Van Valen. Dr. Van Valen is an assistant professor of biology and biological engineering at Caltech. In addition to his training as an applied physicist, he is also, he did his MD training at UCLA. So great kind of connection to Southern California all around. And his lab focuses on the intersection of machine learning, cell biology, spatial genomics to understand cellular behavior, host virus interactions, and disease phenotypes. And I think, what really interested kind of us in his technology for our discussion today is, how these applications, can impact how we think about, a range of disease types, both rare disease and beyond. So David, I'll pass it over to you.
Thank you. Thank you so much for the kind introduction. I grew up on the East Coast, but I was born in Los Angeles, and having done graduate school and medical school here, I think I realized not too long ago that this is home. So I'm very happy to be back and running a research lab at Caltech. And today I'm just going to tell you a little bit about some of the work that my lab does at this intersection of artificial intelligence and biological imaging. And so recent advances in imaging methods and then also machine learning are really transforming how we're able to understand the workings of living matter. So on the imaging side, there are new technologies which are loosely called spatial genomics that provide information on both the parts list variation and then also the spatial variation of tissues. And so, for example, now you can measure the expression and localization of either dozens of different proteins all at the same time, or quite literally hundreds to thousands of genes all at the same time as well. While we've had these advances in measurements, we've also had advances in deep learning. And what these are letting us do is to go into this new imaging data and then pull out meaningful pieces of information with both a resolution, a robustness, and accuracy that was previously impossible. And so some of the things that you can now do with imaging data, I would say fairly routinely, using deep learning methods are shown here on the slide. So, things like identifying cells in tissues, identifying spots in single molecule imaging data, interpreting label-free imaging, and then also restoring noisy images. And so our lab works at this intersection, and a large part of our focus is building deep learning methods that can interpret these modern imaging data sets. So deep learning is actually different than classical computer vision algorithms, and this is actually like what I mean by the title of my talk, Software 2.0. Having algorithms in and of themselves is not enough to get deep learning to work. Most deep learning methods require data, and particularly labeled data, to learn how to perform like their tasks. So this is sort of shown here on this slide. So for example, if you wanted to create a deep learning algorithm that could go into imaging data and then identify cells, the very first thing you need to do is to create a labeled data set, where you have the images that are representative of what you want this algorithm to be able to perform on. And then you'd also need to have corresponding labeled images, where somebody had gone through and then identified, okay, this part of the image belongs to cell number 1, this part belongs to cell number 2, 3, 4, et cetera, et cetera. Once you have that labeled data, then you can go into your algorithm toolkit, pick a deep learning model, train that model to perform this task. And then once it's trained, you can set it loose on new data sets. And so in a very real way, the data is the algorithm. And so a big challenge in this space is, compiling, curating, and labeling the imaging data sets that will get the algorithms to do the things we want them to do. And so as part of that, I would say like a very active effort in my lab just focuses on how much information can we pull out of images using artificial intelligence. And I'd say an example of what we're trying to get these AI methods to do is shown here on the slide. So here we have a classic movie from cell biology, right? It's a neutrophil undergoing chemotaxis, hunting down these bacterial cells, and then eventually it finds the bacteria, engulfs it and eat it. And what we're trying to do is get the AIs, to understand what's happening in these movies in a way that's similar to the narratives that humans create when they're looking at this data, right? And so just things like, notions about like what's a cell, notions about the differences between the red blood cells that are present in this image, the neutrophils, and then also the bacteria. The AI needs to understand the differences between these notions. But that's not all, right? It also would need to understand the interactions between these entities, right? It needs to understand that there's this interaction between the bacteria and the neutrophils called chemotaxis, and it also need to understand that at the end of the movie, there's this interaction called phagocytosis between the neutrophil and the bacteria, and that at the end of all of this, the bacteria ends up dying as well. So this is more of like a longer term, like 5-year vision, but this is the type of resolution that we're trying to get these AI models to pull out of biological imaging data. So with that said, I'd like to tell you a little bit about one of the success stories that we've had, which is going into tissue imaging data and performing single cell analysis. So single cell analysis is a common challenge for modern tissue imaging experiments. As I mentioned before, there are new platforms, imaging platforms that are available that let you get both the parts list and then also the spatial variation within tissues. So a schematic of one of these here is shown here, multiplex ion beam imaging, which effectively lets you measure antibody panels that have on the order of like 30 to 40 different members. You can stain tissues and then image all at the same time. And so what you get at the end of these experiments are N color images where N is approximately 30 to 40, and each color corresponds to some biologically meaningful variable. These are very rich data sets, and I'd say this is particularly important for rare diseases because you can just get more information out of fewer samples. So this is data taken from a cohort of patients that had triple negative breast cancer. We're profiling the tumor immune microenvironment. And you can see here, you have 4 patients and then 7 different markers out of the 40 are shown here. What the details of each marker, it's not really important, but just that you can get a lot of information out of this. So for example, CD45 will tell you where immune cells are, EGFR, P53 reports on the state of the different tumor cells, et cetera, et cetera. And so very rich data, but it's very hard to render interpretable. And one of the very first things that that one has to do to make it interpretable is identify cells, right? So you have to go into images like this and say, okay, this part belongs to cell number 1, cell number 2, cell number 3, et cetera, et cetera. This image here is an image of a stain that targets like the cell nuclei. And so this is a major challenge, if you can do that, then you can do things like identify, okay, this is how much signal for each marker I have inside of each cell. You can do things like identifying cell types. You can identify cell states. You can identify spatial patterns. And if you have enough patient level data, you can link some of these patterns to patient outcomes, as I'll show later. And so here we've sort of taken that image and highlighted the different immune cell populations, T cells, B cells, macrophages, et cetera, in different colors. So this is a challenge. And I would say like compounding this is just an issue that this entire field has had, which is that, when we say identify cells, most people prior to like our labs work in this space, we're identifying like the cell nuclei and using that as a proxy for the whole cell, rather than just trying to find the extent of the entire cell. And so this was a practical reality, just given like the limitations of the algorithms that are available, but it is inadequate for a wide variety of reasons. One, most markers that you're imaging are not nuclear, and so your quantification just ends up being inaccurate. But two, there are analyses that you just can't do if you're looking at cell nuclei, right? So one is looking at the subcellular distribution of different markers. Another is looking at cell morphology. And so we set out working with colleagues at Stanford to solve this problem using deep learning. The very first thing that we did is we just compiled data sets, images that were representative of what folks are imaging and the different imaging platforms that are being used. And then what we did is we took these imaging data sets and then labeled them with single cell labels, highlighting both the cell nuclei, but then also like the full extent of the whole cells, the cytoplasms. And just visually speaking, you can just see that this is a hard problem. So for cell morphology varies like quite considerably, both within and across tissues. You'll have differences in autofluorescence characteristics across different imaging platforms. Some tissues will have cells being sparse, others will have cells being dense, and then some tissues, most notably the GI tissues, a number of cells are just missing cell nuclei, and this is an artifact of taking a two-dimensional cross-section through a three-dimensional object. Labeling is a big challenge, and so we actually ended up creating our own labeling software and labeling framework exactly for this form of data. And so we've created this browser-native software called Deep Cell Label, which lets us create single-cell labels for these form of data. We can also label things like cell types. And a very nice feature about this software is it's browser native. And so now we're not limited to the folks in lab doing labeling. We can actually harness pools of crowdsourced workers in other countries. And we've come up with efficient schemes of labeling, where we can leverage, what these crowds can do, what the human experts can do, and then also what the AI models can do to collaboratively build these data sets rather than just having experts crank away in a linear fashion. And so with this software, we've been able, and crowdsourcing, we've been able to label a large amount of tissue imaging data. And so we recently released this data set called TissueNet, which is meant to be like a resource for multiplex tissue imaging. It contains approximately 1.2 million whole cell labels and a comparable number of nuclear labels as well, collected. Sorry, I think it's my slides are set to like go to automatically go forward. My apologies. But it's collected across approximately six different imaging platforms, 9 different tissue types, 3 different disease states, histologically normal, cancer, and infectious, 3 different species, mouse, human, and macaques. And because we were using this collaborative form of labeling rather than having experts labeled, it took about 4,000 person hours, or so about 2 person years worth of work to label all this data. But because of the way we're labeling, most of this was done in a distributed way by crowdsourced workers, and the experts actually had to contribute relatively modest amounts of time. And so with these data, we can then train deep learning models to identify both the nuclei and then the whole cell. And so I can just show you what some of these results look like. So here's a image from a pancreatic ductal adenocarcinoma, and then we can run it through our algorithm, which we call Mesmer, and you can see like these white outlines of predictions for the boundaries of whole cells. The really nice thing about this algorithm is that it generalizes across all of these diverse tissue types and imaging platforms and disease states. And so here we see like a collection of different images. And then here we see the predictions produced by our algorithm Mesmer, along with the F1 score, which is a measure of segmentation accuracy. And you can just see visually that Mesmer does like a really good job across all these diverse imaging data sets. It does so well, in fact, that when we compare it to human performance, groups, we actually don't find a difference. And so this is data showing a comparison that we did where we asked pathologists in a blinded fashion, which do you prefer, the outputs of our AI model or the outputs of what human labelers would produce. And we saw that across a wide variety of different imaging data, the pathologists didn't really have a strong preference one way or the other. And so this is sort of demonstrating that this algorithm we've been able to make can perform these tasks with human level accuracy. And just to highlight, it's the diverse training data that we generated that's driving this performance. Because we have this algorithm now, now we can, automate the analysis of tissue imaging data, and a lot of the subsequent analysis steps become much easier. So, for example, identifying cell types in these data sets. We've been able to show that with our segmentation algorithm, we're able to do this it's much faster than if you're relying on segmentation that's inaccurate. And then analyses like looking at subcellular localization of different protein markers, or looking at cell morphology, we can look at those in a fairly automated fashion too, and these are also accurate. With this ability, we've also been able to demonstrate that we can actually tie some of these imaging features to patient outcomes, and so this is data from a collaborative work that we did with Liat Karen and Michelangelo at Stanford, looking at patients with triple negative breast cancer, subjecting their tumors to this sort of spatial profiling. And what we're able to find is that there were actually different dominant themes of spatial organization of the immune cells across these different patients. 3 different patterns we identified: the cold, infiltrating, compartmentalized. Infiltrating, the immune cells are all intermixed. The compartmentalized, the immune cells form their own compartment. And because we had outcomes data, we're actually able to look and ask, like, okay, are there differences in outcomes among these patients? And between the infiltrating and the compartmentalized, there actually is. The patients who have a compartmentalized phenotype actually do better than the ones that have infiltrating. And I just want to highlight, these are relatively small cohorts. So this is on the order of like 30 different patients. But because we're doing this deep profiling and this like very accurate analysis, we're actually able to find like these sort of signatures, even though we have small numbers of patients. And so with that, I will stop talking and open the floor for questions. A huge thanks to my lab and collaborators who actually did the work. And then, we're very grateful for the funding we've been able to procure for these efforts.
Great, thank you so much. I'll open it up to any questions. Maybe just to kick it off, and it reminds me, I think with Roger Perlmutter's new company, Icon, he had this great kind of quote that was like, if you can't see it, you can't understand it, and you can't treat it, right?
Yes.
So I would just love to know, how are you thinking about, there's just so many applications in terms of how this could translate, whether it's on the drug discovery side, whether it's on, molecular diagnostics. What are some of the kind of key translational applications that you've kind of, started thinking through in terms of, how this can kind of, take that step towards, being part of the pipeline of new potential kind of translational technologies?
So I would say let me stop sharing my screen. I can reshare it when if folks want to see slides, but it's helpful to see faces when I'm answering questions. So I'd say we've thought quite a good deal about commercializing this technology, and so I put my answer sort of in the context of commercialization. In my view, I'd say there's sort of 3 or 4 different avenues that one can pursue. So one is more of a sort of a platform play. So these sort of profiling technologies are very useful for profiling, immune cell behaviors, which, as you can imagine, are very important across a wide variety of different therapeutic plays, right? And so if you're so one would be a platform type play, where if you're a company, engaging in a clinical trial where say you're evaluating a CAR T cell or some immune checkpoint marker in solid tumors, and you want to sort of do this sort of multimodal profiling to identify, okay, who's doing well, who's not doing well, is there anything that's predictive adverse events? These algorithms and this type of spatial profiling is can be very useful. And I'd say one of the hypotheses that's been present in this space, at least, is that it's spatial interactions that are sort of dictating among different cell types that are sort of dictating patient outcomes. And so this would be like an enabling technology for those sorts of players. The second sort of play would be more like a therapeutics play, right? Sort of using these platforms, these measurements, and the fact that you can pull information out of these measurements as part of like a drug discovery effort, right? And so there is more like target discovery, like what new targets can you identify, and then how can you tie them to like disease, sort of disease phenotypes? What can you do with these sort of technologies? There, I would say like the technology case is a little bit trickier, because there's when you think about like the different, when you think about how you'd use these spatial and also these omics technologies in sort of a drug discovery effort, there's no one assay that actually gives you everything that you would need, right? So for example, the spatial proteomics, amazing technologies that let you go through like FFPE tissues. Their downside is that you have to know what you're looking for a priori, right? Because you have to like design these antibody panels, do your staining, et cetera, et cetera. Some of the methods that, don't need that a priori knowledge, so like the transcriptomics or spatial transcriptomics, they also have their own issues and their own limitations, right? The transcriptomics in general, they work much better with fresh or frozen tissues rather than the formalin fixed paraffin embedded. So you're right off the bat like limited as far as like your samples they have available to you. And then as far as the spatial transcriptomics methods that do not need sort of a priori knowledge, those have not reached like single cell resolution yet, right? And so the resolution as far as like, okay, what cell types are present, like how are they interacting and whatnot, you don't quite have that. And so for that sort of like discovery effort, really what I would say is like this would be part of like a portfolio of methods that you would apply to a particular disease problem or disease phenotype. And then I'd say like the third potential application, sort of related with like issue one, are spatial biomarkers, right? So finding like spatial markers of disease that help you like stratify patients. A very nice example was a paper recently published by my colleague Michelangelo. Right, they used these algorithms as part of like their work, but they're looking at patients who have ductal carcinoma in situ, precursor lesion for breast cancer, and they want to understand, okay, who's going to progress, who's not going to progress, right? And so using this sort of like deep profiling combined with the AI, they were able to identify some spatial features that were predictive as far as like which DCIS patients are going to go on and then have invasive cancer. And so thinking about identifying spatial biomarkers across different disease states, I think is something is another potential area where these methods can have like translational impact.
Yeah, definitely like kind of a step beyond kind of our, histopathology, which it seems to me has been around for
it's a step
5
beyond.
decades.
I completely agree.
Multiple steps beyond.
Yeah, so that's I'd say like the very interesting thing about these methods is like how they get you like the information, right? And so the way I view it is histopathology. The information is present in like the pathologist's brain as far as like, okay, you're looking at H&E image, what does a particular cell type look like, B cell, T cell, tumor, macrophage, et cetera, et cetera. And, they're not 100% accurate, but, accurate around, 70-80%. And, you can ask pathologists to, sort of label these sorts of images for you. Right, that's something like Path AI did to like to pretty good effect. The spatial profiling methods, like you get that information, that same type of information. Oftentimes you can get it with like even higher resolution, but it comes in the price of your measurement, right? So now instead of relying on what a pathologist can tell you, now you're relying on like what an antibody can pull out of your tissue, or what an RNA fish probe can pull out of your tissue. So there's a trade-off, right? Because the H&Es are very readily available, easy to acquire. This deep spatial profiling is harder to do, substantially more expensive. If you're doing like a Pyle experiment, for example, you're looking at already like a six-figure sum as far as like reagents and then instrumentation time. But you don't need to rely on like what a quote unquote expert could tell you, right? The algorithms can give you that information.
Yeah. Martin, go ahead. I see your hand up.
Yeah, great talk, David. Very interesting work. I was curious on the translational aspect and commercialization. Could you comment on the IP that you're pursuing here? Is it really just copyright, or are you looking at patents on algorithms? What might be the foundation of the intellectual property?
Yeah, so the foundation of the, so there's two things. So there's, well, three things. There's a software infrastructure, right? So we have like this cloud native infrastructure for developing and deploying like these AI tools. It's built on Google's cloud engine, but we've done it in such a way where we don't have to pay like the exorbitant costs of like a managed service, and so we can enjoy like a low price for inference. There's the algorithm itself. So the deep learning algorithm that you train on like your training data set. I would argue that while at the time it was like fairly novel, novelty as far as like the algorithms is rapidly diminishing, and actually like in this space, academia is not where the most innovative algorithms are coming from. Most of them are coming from either technology companies or the companies engaged in like the self-driving car efforts, because there, you really need to have like the fidelity. And then there's a third thing, which is where the actual like moat comes, and that's the data. Right? If you do not have the training data set, then you cannot build an algorithm that's able to perform with this type of accuracy. And that is really what gives the moat for these sorts of the technology I displayed today. So now, as far as like a commercialization effort, where does the actual like protection exist? In my view, it's the data, and then like the software systems that we've built. The data, it's a very powerful resource. As I showed, like it would take, like if somebody wanted to do it again, it'd take about like 2 years of work to do it. That's likely like not, it's not impossible for somebody like say like 10x Genomics to take like $5 million and say, okay, we're going to build something that can do like the exact same thing. It's feasible, and there are companies like, for example, Appen, where they have like crowdsourced workers, they gladly take your $3 or $4 million to go do this. The real moat comes in our capability to generate new AIs. Because I said, if you want to incorporate this as part of a discovery effort, what I presented today is like, building block number 0 out of like the 5 or 6 or 7 different AI pieces that you would need to actually like make that kind of endeavor successful. And there, what we built is just not enough. You need algorithms for doing cell segmentation, you need algorithms for doing cell type identification, you need algorithms for identifying spatial patterns, you need algorithms that can operate on both the spatial data sets and the non-spatial data sets. It's not a small list. But because we built like this software ecosystem for developing like these AI models in an integrated collaborative way, where it's not, humans doing everything, it's humans plus AI. And then if you need help from like people outside, you can bring them in. Because we built this platform, for us, it's actually like much cheaper to build these models than quite literally like anybody else. And there, I think like that's like the real like moat that we have as far as like protecting the IP. And there, we've been doing like our due diligence. All of our software, although we're active in this open source community efforts, everything is licensed so that free for nonprofit. It's for profit. You need to seek a license. If anybody asks for a license right now, we tell them no, because we are actively working to commercialize this. But we've thought a good deal. I'd say a problem in this space is that the tools that I'm presenting now, that I presented today, are sort of like an externality, right? Like they enable everybody to do cool, innovative things, but nobody really wants to pay for them until like you have them and they're taken away, right? So example like roads, right? Everybody loves roads. Nobody likes a tax bill for building like new roads or infrastructure. And so part of what we're thinking about for commercialization is to maximize the value for the investment we've made already in building these tools. It does need to be paired with some other like discovery, some other effort, right? So either spatial biomarkers or drug discovery effort.
Well, thank you so much. So I think, I'll go ahead and transition to Dr. Kimonis, who's going to drive us home today. So Dr. Kimonis is a clinician scientist and professor in the Division of Genetics and Genomic Medicine and the Department of Pediatrics at UC Irvine, also Children's Hospital in Orange County. Dr. Kimonis specializes in the diagnosis and management of children and adults with neuromuscular, neurodegenerative, and dysmorphic diseases. She has a really active clinical research and lab, and focuses on a number of disorders, particularly lysosomal storage disease. She also actually, this relates to some of Alysson's work, has a registry focused on lysosomal diseases. So Dr. Kimonis, I will pass it over to you.
Thank you so much for the kind introduction. So today's title is Translational. Can you hear me okay?
Yes, we can hear you. Great.
Okay, great. Translational Studies in Rare Genetic Disorders. And as you mentioned, I'm at UC Irvine. I'm a clinician scientist, so I also see patients, and that's a problem because there's never enough time for research. So my interest in this rare disorder that you will learn more about stemmed from when I was an assistant professor in 1998. It's a long time ago, and it's embarrassing that I don't have a treatment for this condition as yet, but working on it. So this family were diagnosed as a unique family with autosomal dominant limb-girdle muscular dystrophy and Paget disease of bone. And I had learned to map genes when I was at the NIH as a trainee, and we actually mapped the gene to chromosome 9, where VCP was identified as the gene responsible for this condition. The first paper only had 6 mutations, but since then there have been over 60 mutations identified. Most of the mutations are in this N-terminal ubiquitin binding domain, as you see over here, but there are also mutations scattered elsewhere, but less so in the D2 and D1 domains. And there is some sort of phenotype correlation in that the A232 mutation, which is in this D1 domain, has been noted as the most severe of all. So
Dr. Kimonis, are you supposed to be sharing slides at the moment? We don't see your slides yet.
I am sharing slides, so is it not moving? Oh dear.
We're not seeing the share slides. Yeah.
I'm sorry. Okay, I didn't realize. Okay, my apologies. Okay, so let me go back to that. Okay, so let me swap slides.
All right, we can see them in presenter mode now. So
yeah, I'm trying to figure out
step in the right direction.
How to get it to the other mode. Hide presenter view, but that's not what I want. Maybe I can just start over. It's always a challenge when you're using 2 screens.
I think also if you just, we could see it okay. I mean, I think if it's in presenter mode, that's fine.
Let me go back there. Okay, is that okay?
That looks great.
Yeah, it looks great.
You can see it. Okay, sorry about that. So this was that first family that I mentioned, and then this is the gene with the mutations in the N-terminal domain. And then this is the gene. So it's the VCP protein. It's a triple ATPase, and it's important for many functions. There are 2 catalytic domains, and D2 provides most of the ATPase activity. So what the protein does, it interacts with a bunch of cofactors and with its binding partners. It sort of pulls in proteins destined for ubiquitination and proteasome degradation through that central pore, and it does it through the ATPase as the driver of this process. What we know about the mutations is that they are caused by gain of function. They're all missense, resulting in gain of function. So the main phenotype pertains to inclusion body myopathy. 90% have this, and individuals become weak in their early 30s. This gentleman is not able to raise his arms up. He's being lifted, his arms are lifted on the backs of chairs. And there is deltoid muscle wasting, there's marked scapular winging. And when you look at the muscle, there are these rimmed vacuoles with the arrows and showing, and there are inclusions that are stain positive with ubiquitin. TDP, autophagy markers, P62, and the VCP as well. Paget's bone disease is seen in half the patients, and really we have no clue why half don't get it and why half get it. There is some theory that if you are exposed to measles, that those viruses remain and are the nidus of these Paget foci occurring. And we know that the incidence in the general population is decreasing because of measles vaccine, but that is not the case with this disease. Approximately half get it. 30% get frontotemporal dementia, and there is significant atrophy of the cortex, but particularly in the frontal temporal region. The temporal region is which controls a language, so, and you get accumulation of TDP, ubiquitin, and caspase. 10% develop amyotrophic lateral sclerosis. Sometimes that's the only feature. If you look at families with familial ALS, 1 to 2% are accounted for by VCP. So these are families without the syndrome that I have shared with you. So at the beginning, I was not a researcher. I was purely a clinician, but nobody was interested in rare diseases. And so I learned how to do lab work, and the initial mapping of the gene, finding the gene was not sufficient, and we learned how to make mouse models and develop myoblasts for translational studies. So these are some of the cell lines that we work on. Unfortunately, primary muscle lines have a very limited lifespan. You can't keep taking muscle samples from these patients. Besides, their muscle turns to fat and fibrous tissue, and you expose them to painful procedures, and at the end of the day, you get nothing. But these iPS-derived myoblasts have been a great model, and people like Pyle have been pioneers in making it easy for us, easier. I mean, it's never easy. What you see over here is the control lines on the left and the patient myogenic lines on the right. And in this disease and similar disorders, you find that there is TDP43 in the cytoplasm and not in the nucleus, as you see in the control. And there's increase in autophagy markers, LC3 and P62. And what you see on the right, E, is the Western blot showing that there is TDP in the cytoplasm, which I mentioned was the hallmark. So I mentioned that we learned to make a mouse model. This is a knock-in mouse model, which we surprisingly are still using in the lab 10 years later. So this is where the mutation is, and we made pre-log sites in order to have the facility to actually remove this. I'm not showing you the slides of what happened when you remove it, but we actually found that the phenotype had improved. So when you look at these heterozygote mice, patients are heterozygote, they do develop a little bit of weakness. There is some muscle pathology, and there's cytoplasmic accumulation of TDP43 and ubiquitin positive bodies, which also stain, and there's increased autophagy markers. The brain, spinal cord are also involved in the pathology. So very similar to the humans. Unfortunately, this mouse model as a heterozygote is not a terribly useful mouse model. So we are working with other mouse models and the homozygote with this knock-in. So the homozygotes you can see on the right, they're smaller, they don't move, and they live only 3 weeks, and they have very marked pathology. At the top of each panel is the wild type, and then you see that there is disintegration basically of the muscle, and you see at the bottom panel where the mitochondria are very bizarre, large, misshapen, very disrupted, crispy formation. I mentioned the life survival curve. They only live 3 weeks, but what we serendipitously found that if you feed the pregnant dams a high-fat diet, these mice are now living. So those are on the normal diet, and these are on the high-fat diet, just a little bit more, 50% more fat, and it's mainly based on soybean oil, and that's the difference over here, the increase of the child components. So when we looked at the myoblasts and the patient myoblasts as a result of this finding, we found that there was more ceramide in the mutants, and we have used different strategies to actually reduce the amount of ceramide. Myriocin R14494, which was invented by Dr. Piomelli, L-cycloserine, and then this RNO82 is a positive control. It increases the amount of ceramide, and indeed it does make things worse. But we saw improvement in the TDP that you see here. Also, the LC3B improved a little bit, the ubiquitin. This was published last year. So I mentioned to you gain of function as a result of the mutations that cause this disease. So we're looking at many different strategies to reduce this gain of function. In particular, we published a VCP inhibitor, CD5083. We're currently studying antisense oligos and using gene therapy strategies to get in these strategies into the muscle. So this was a very important paper that told us that we were on the right track. So that's the wild type, and these are the mutants treated with Drosophila mutation treated with DMSO versus the earlier VCP inhibitors. And what you're seeing here is restoration of the mitochondrial pathology to that of a wild type. And we studied CD5083, which is a product made by Cleve Biosciences. They are based in Northern California, and they had conducted a study in about 85 people with solid cancers. But unfortunately, the feds stopped their study because of visual problems, which we did not actually see at the doses we used, because we were not wanting to knock down cancer. We're just wanting to downregulate VCP because of, this is a VCP inhibitor. We didn't want to totally knock down VCP. So what you see here is when you treat the myoblasts with different doses of the CD5083, you're seeing amelioration of the TDP43 pathology. But we actually saw an increase of LC3, and we know this drug actually increases autophagy, which is not a bad thing, because autophagy, increased autophagy actually removes protein that's destined for degradation. And this is, looking at the cytoplasmic quantification of what I just showed you. We also treated our mice. I mentioned to you the heterozygotes are not really very useful model anymore, but the homozygotes showed improvement with the treatment. We saw amelioration of the TDP pathology as well. These are just quantifications at the bottom. So I mentioned antisense oligo. So we have partnered with Ionis, and they have generated antisense oligos. And the patient support group was also very helpful in actually forging this relationship with the company and our lab. And we have worked with 3 different ASOs. ASO2 seems to be the best in knocking down VCP. And we have an ongoing study in a bigger cohort of mice to look for differences in the phenotype. So I mentioned to you ways of knocking down the VCP, but in order to get into the muscle, we need a more robust strategy. And we're looking at myo AAV and AAV myo as a way of getting in the inhibitors of VCP. So what you're seeing here is that VCP inhibitors will knock down VCP. It does not discriminate between the mutant and the wild type. And what we're planning on doing is actually give VCP, normal VCP, and then the combination hopefully will work better. So we knock down the mutant overall, and then give wild type VCP, and we hope that the cells and the mice will enjoy that more. So we're waiting for some of the constructs to come, and hopefully we'll be able to see dramatic improvements. This is just our preliminary study looking at AAV myo, and compared to AAV9, there is very good expression in the muscle through all these different muscle types that you see here. So this is very exciting that we can target muscle. So this is a summary of what I shared with the VCP related studies that we're working on in the lab. I'd like to very briefly tell you about some of our other studies. So because of my interest in neuromuscular disorders, we are working on Pompe disease mice, as well as another very similar disorder clinically to VCP associated with point mutations in the HSPB8. And I'll tell you very briefly about the NUBPL project. So Pompe disease is a neuromuscular disorder. It's a glycogen storage disorder. And what happens is that with deficiency of this alpha glucosidase, GAA, you get accumulation of glycogen, which normally would be converted to glucose. And so you get accumulation in the muscle and the heart, the intima, and they die early from muscle weakness. And the strategy we're using, and this is Ionis, partnership with Ionis, we have made GYS1, which is a muscle-specific glycogen synthase, antisense oligos, and the rationale for that is if you knock down glycogen synthase, you won't make glycogen. And then this is a way of using substrate inhibitor therapy. And then whatever remaining residual GA activity the person has, because there are many more people with the adult onset type where they have up to 30% enzyme activity, whereas the infantile type have almost zero enzyme activity. And we expect that this glycogen will be degraded because now there's less for it to be degraded, less for degradation. So the antisense oligo is being used, and it inhibits translation. And we hope that there was no glycogen synthase being produced and reduced glycogen. So some of the initial work done by Ionis with ASO1 and 2 and then 3 showed that there was a downregulation of the GYS1 muscle specific. And what we have done in our lab is treated with either ASO1, 2, and 3 and looked at the results. We found that ASO3 was the most effective in knocking down GYS1 and reducing glycogen. And then we followed that with enzyme replacement therapy. Because these are null mice, they have no GA activity. You have to actually immunomodulate them with a diphenhydramine, otherwise they will get horrible reactions. And we inject them with this enzyme replacement through their tail vein. And then this is the results, what you see here in the quadriceps heart and the diaphragm. And then you see that's the control PBS, and then these are the mice treated with ASO3, ASO and enzyme replacement and enzyme replacement. And you can see that there is really dramatic downregulation of glycogen synthase activity across the tissue. And then when you look at glycogen, it shows that there is improvement of glycogen, and the improvement is much more marked when you combine it with enzyme replacement therapy. And the same thing with not glycogen. And this is rotarod activity. So at the bottom you see here is untreated, and then at the top you're seeing either treatment with ERT or ASO. And it's just a small cohort of mice. There were just 5 in each group. And we think overall that if you look at the endpoint at the 7-month mark, that the ASO and ERT seem to be better, although the ASO on its own, the earlier period appears to be effective. But at least this needs to be repeated. I'd like
Dr. Kimonis, maybe just to jump in, I just want to make sure there's enough time for questions.
Sure.
We're getting close, but if you have a couple more slides, go ahead and wrap up. I just want to make sure that we can get in a few questions before we wrap here, since I know we're.
I just wanted to note that we're working on this other muscle disease with frameshift mutation. They're very similar to VCP. Clinically, we've made myoblasts, and they have a phenotype that's there's a TDP and autophagy accumulation, where We made a mouse model and we're finding similar pathology. There's weakness of the animal, and we're interested in looking at different treatments. So we've used one treatment, colchicine, which improves HSPV8, but unfortunately the patient didn't do well. And then just the last slide, NUBPL is a mitochondrial complex 1 disorder. Patients have absent cerebellums, they have cognitive deficits, and we're working with a company to try and develop antisense oligos to correct this mutation. So that's it. We made a mouse model, but unfortunately the homozygotes don't survive. And you know, we as a clinician, I'm seeing so many different patients, and there's a huge unmet need, and it would be wonderful if when I see a patient, I can say, well, you know, there is a potential treatment from CRISPR or gene therapy or whatever, and you'll be able to help them. So of course, I have to show my acknowledgement of all the people who did the work and the funding. Thank you for the opportunity.
Great. Thank you so much, Dr. Kimonis. And I know, I think we were scheduled to go to 11:45. For those of you who want to stay on and just ask a couple questions, you know, want to be sensitive to time. I had one, and you know, that was with the VCP myopathy. You know, one thing that I found interesting is the TDP-43. We're just seeing that everywhere, right? You know, ALS, other forms of frontotemporal dementia. There's companies that are going after TDP-43, either, you know, via ASOs or small molecules. I just, you know, on the biology side, you know, how much of that do you think is truly, you know, I don't know, kind of a disease, you know, is causal versus, you know, kind of a, you know, more of just kind of the pathology of something more underlying? How do you think about that marker in particular?
No, that's a really good point you're making, because the pathology is so similar to ALS, frontotemporal dementia, and if we can find common targets, it will help a whole host of these disorders, which are not rare. You know, these disorders that each of the different components that make up this rare disorder is actually quite common in the population. So studying this rare disorder has huge implications for common disorders.
Great. Any other questions from those? Otherwise, we'll go ahead and wrap it up here. I really appreciate everyone's time today.
Thank you.
Excellent.
Thank you for the.
Thank you so much, everyone. Have a good rest of the week.
Yeah, thank you, everybody. We'll be posting the recording in the next day or so, and really appreciate everyone's time today. And of course, we'll connect you all with the emails and contact information so all you guys can connect after today. So thank you very much.
Thank you.
Thank you. Bye.
Bye.